So here's our fourth part in the How Cells Divide lecture. So think about what we've covered so far. Prokaryotic cells divide through the process of binary fission. So bacterial cells, archaea, simple process, one cell copies its DNA, gets divided in half, and now you have two identical, or what should be identical cells. Quick, fast, 20-minute process in some bacteria, and it allows for rapid reproduction. Ideally, everything's identical, but bacteria make mistakes, and this is what leads to new variations in those particular organisms. Now, the eukaryotic cells are the ones I want you guys to primarily concern yourselves with. Plants, animals, how do these cells replicate? And that is through the cell cycle and that process we've been talking about called mitosis. So keep in mind with the cell cycle, you have interphase, which involves G1, S, and G2, and then you have mitosis, which is composed of prophase, prometaphase, metaphase, anaphase, and telophase. Very ordered series of events that allow the cell to divide itself and create two identical cells. So those cells are supposed to be used for growth, repair, healing, all those factors that we need that are necessary to sustain life for a multicellular organism. Now, when the cell is going through the cycle, it will have to go through a series of checkpoints. This is just like going to the airport and taking a flight. So if you think about flying, go down to the bottom right corner of the screen here, down here. Your first checkpoint is when you enter the airport and you have to get your ticket. You go up to the ticket counter, they're looking for an ID, they're trying to make sure it matches. Is everything correct? Are you on the right flight? Are you at the right airport? Is this really you? Provided you pass those questions or you establish that this is where you need to be and you're at the right spot, you move through the checkpoint. You then go through the airport, you hit security. It's like the second checkpoint of the cell cycle. Let's check to make sure you're going the right direction, you have the right information, you're valid to be here. If everything's good, keep going. Third checkpoint is before you board that plane. You're at the gate. One last time, let's look at your ID and make sure it really is you and that you're in the right spot. If all is good, get on the plane. The cell does the same thing. There are 3 checkpoints in the cell cycle. So the first checkpoint, we could say, happens in G1. The cell evaluates the condition of the cell. Is it growing correctly? Is it making copies of cellular organelles? Is the size reaching optimal size? Does the cell have the energy and the nutritional factors it needs to sustain and support this growth and this cell? If all things are good, it moves forward. If not, the cell can do self-destruct and eliminate it. Or the cell could fix problems. Maybe it needs to wait a little longer before it grows more. Maybe it needs to draw in more nutrients, and it's not going to replicate as rapidly as we want, but it's going to do it, hopefully, in a better fashion and make sure everything is accurate. The cell goes into the S phase, copies its DNA, moves into the G2 phase, and here's where it's going to look at the DNA and say, did we do this right? This is like the security checkpoint. Did we make this copy correct? Because if there's a problem in your DNA, every cell that you create after this point forward is going to be incorrect. It's going to be different. It's going to carry that flaw in the genetic material. Now, depending upon where the flaw is at, that could be completely benign and have no consequence at all, or it could cause the cell to have significant problems. So let's check it in G2. If all is good, you move on. If things are bad, you either fix it or you self-destruct. Assumably, let's move forward to the M phase, the mitosis part of the cell's life, specifically metaphase. The cell's going to look at, are all those chromosomes in a line? Are they all lined up on the what we call the equatorial plane or across the center of the cell? Is everything positioned correctly? If so, continue and go into anaphase, telophase, 2 new cells. Let's do this again. If there's a problem with the chromosome positions, can we fix it or do we self-destruct? So the cell is constantly doing this. Every single time it goes through that cell cycle, it's trying to evaluate the quality of the cell. So if the checkpoints are functional, then the cell moves forward. It continues to go through that cell cycle and it will progress to the next stage. So all of us have checkpoints. There's a particular protein called the p53 protein that acts as the checkpoint person or checkpoint structure. It's like that security guard, it's the person at the check-in counter at the airport, or it's the person at the gate when you're actually getting ready to get on the plane. A normal p53 protein is a gene. That gene allows for repair to the DNA, fixing things, correcting things, and making sure everything goes well. But if the DNA is messed up, if your p53 gene isn't working, then we can have a problem. But again, the key is that gene acts as a checkpoint regulator. So it's watching, it's looking, and the p53 gene can actually trigger the destruction of the cells if the cells are too far damaged. So it's going to, like the rocket launcher there, it's going to pull that trigger and destroy the cell if it cannot be repaired and it cannot be saved. So we all have this gene. The key is your gene turned on or your gene turned off? Now, if the checkpoints are not functional, then the cell may continue through the process, but they will produce abnormal cells. So imagine your security guards are sleeping, and you walk up to the gate, you go right through security, they don't check anything, you're on the wrong plane. Well, same thing can happen in our cell. If the p53 gene, that protein, is not correct, it misses the mistakes. It allows a cell that is different to continue through the cell cycle. So when you look at the abnormal p53 gene, doesn't catch this mistake here. It fails to stop the cell division and fix it and repair it. So this cell goes through that checkpoint, it divides, and eventually it produces 2 copies of itself that are not correct. Those then go through and divide again and again and again, and this is where the problem of cancer lies. For many people, the p53 gene is not functional. It's not working, and your cells go through the cell cycle and they never stop. They're behaving abnormally, they're not following the rules, and it leads to, unfortunately, all those problems with cancer. So cancer is basically when cells go bad. They're acting in a different fashion than they should. Now, sadly, 1 out of 3 people will develop some form of cancer during the course of their life. So here are some statistics about cancer. 1 out of 13 people will develop lung cancer. 1 out of 7 women, primarily women, will develop breast cancer. Men can get it, definitely not that common in men, but 1 out of 7 women, unfortunately, will develop breast cancer. Skin cancer, about 1 out of 6. Prostate cancer, male specific here, roughly 1 out of 6. These are the rough statistics. The numbers are changing with breast cancer awareness, with prostate cancer awareness, with all the awareness we have about cancer. Those numbers are starting to change, which is a great thing. But I encourage you guys, look at your family trees. We'll do this more when we get to genetics, but start thinking about your families. If cancer is in your family, is there a genetic connection? All right, so here are the factors that increase your chances of getting it. Start with your genetics. If mom had cancer and dad had cancer and grandpa and your uncle and your brother, odds are your genes, your checkpoint genes, are turned off. They're not catching mistakes. They're unfortunately not working. And there's nothing we can do about that at this point. Are we exposing ourselves to radiation? X-rays. Those of you heading to healthcare, you don't stand in front of the X-ray machine. That's what those lead vests and the shields are for. We don't expose ourselves to ultraviolet light, sun, things like that. Laying out during the summer increases your chances of getting cancer. Different organic chemicals, different viruses, all of these things have been linked to causing cancer. So we want to look at our genetics and the environment to determine, are we increasing our odds of getting cancer? So the scary statistics, 1 out of 6 for skin cancer. If you have 6 people in your family, 1 person statistically is going to get skin cancer. And if it's not caught early, it can spread because it doesn't ever stop going through the cell cycle. Cancer cells do not die after 50 spins of the cell cycle. They go on and on and on and continue to constantly cause health problems. If left untreated, there's a certain time where nothing can be done. So I'm going to jump back a slide here. This is a X-ray of the lungs of a person with lung cancer. All of these gray masses that have the purple border, those are tumors. At that point, there's nothing that can be done for that person. If it was caught months earlier and it was one small little spot, your odds of surviving cancer are significantly greater. Early detection is the #1 way to deal with cancer. Catch it early. That's why I have the little image here, the ABCDEs of detecting melanomas. Those are skin cancers. So if you have some kind of weird shaped dark spot with uneven borders that varies in color, might be large, changes shape, etc., that might be something to take a look at. You ignore it, it's not going to go away. It's just going to continue to divide and divide and divide and then spread. At a certain time, cancer cells break apart and then they move through the bloodstream and they start showing up everywhere else. So what may have started as a small melanoma on your shoulder or your nose, next thing you know, you just ignored it for months, and next thing you know, it's in your lymph nodes, it's in your bone marrow, it's elsewhere. So cancer is not something to just brush off. Even when you're young, you don't ignore it. It can unfortunately hit anybody at any age. It does not discriminate based on race, gender, age, etc. It's something that affects everybody. So the good news, here are the statistics. 99% of men diagnosed with prostate cancer will be alive 5 years later if they get treatment. That's phenomenal. But you have to look for it. If you're at high risk, you check more often. 88% of women diagnosed with breast cancer alive 5 years later, if caught early enough. This is the idea of going in for those mammograms, doing those monthly checks. Start with the genetics though. If your sister had it, your mother, your grandmother, your aunt, there is a direct genetic link to these. So I encourage you guys, have that conversation with your parents or grandparents or other family members about their health. It will make a big difference for you. Colorectal, unfortunately, odds decrease. Lung cancer, sadly, is very small. By the time anything's done on lung cancer, there's not much that can be done because of where the cancer is. So early detection is the key to surviving cancer. Now, research is discovering different genes play a huge role in this. Oncogenes are genes that cause cells to become cancerous. These are the genes of tremendous amounts of research trying to discover why they cause us to develop, why this happens, what leads to cancer. One of the main genes we talked about was P53. I want to keep an eye on that, and the other tumor suppressor genes.